in a nutshell, we look at how muscle stem cells, particularly fibroadipoprogenitors or FAPs, are responsible for muscle degeneration in a variety of different states. And as a shoulder surgeon, that mainly comes up in rotator cuff injury. But we also want to see how these properties can be leveraged for muscle repair, because we know when you're young and healthy, these cells are responsible for guiding muscle to regenerate and repair. But as we get older and we have chronic injuries, it just doesn't work that way. We think that by kind of turning on the right cellular machinery and the right genes within these cells, you can leverage them to give the right signals or the right energy to promote muscle recovery in a variety of different situations, whether that's in the setting of rotator cuff repair, low back pain, or even as far away as aging and other neurodegenerative conditions. I knew from a relatively early age that I always wanted to do research. I started doing basic science when I was a first year undergrad. I was working in a lab that was looking at anesthetic agents and stroke, which I don't remember anything about the project, but I recognized that I liked the the kind of the process itself. And then when I started, or when I transferred to Stanford and was fortunate to work with a group of people looking at oncogenes and gene transcription factors and what turned on and off oncogenes, and I thought that was a really interesting, clinically relevant project. So I thought at that point, I was like, I'm pretty sure I want to do research, but I just didn't know what I wanted to do. And then when I was in med school, I continued to work in that lab for a little bit because I stayed in the same spot. And when I was in med school, I transitioned to work with somebody named Bobby Robbins, who is now president of the University of Arizona. But at that point, he ran a cardiothoracic surgery lab at Stanford where we looked at ischemia, reperfusion injury in transplants. And what I really liked about working in that lab was the fact that he allowed us to kind of explore our own hypotheses within the envelope of asking the same general question. So it gave us a lot of independence. And then once I decided I didn't want to be a cardiac surgeon for a variety of different reasons, I was fortunate to go to UCLA and work with Jay Lieberman, and he had a very similar philosophy of supporting us primarily as independent researchers. And again, if it fit under the general principles of gene therapy or looking at things that can promote bone healing, we were allowed to do what we wanted. And that was really helpful in terms of allowing me to decide what I wanted to do. And I really try to run my lab now and how those two ran their lab. I think there's a bunch that come to mind. Some are bigger, some are smaller. I think the big one for anybody doing research is always funding. None of us have an unending amount of funding, although was never quite sure what the funding was like at HSS. That seemed a little bit suspect. They seemed to just have money. But I think at the start of my career, the question was always whether or not I would have funding, not where would it come from. It was like, could it actually happen? And particularly now with a much bigger group, our lab right now is somewhere between 10 and 20 people, depending on what part of the year it is. It's hard to find enough money to do all the things that we want to do, and then make sure that that's sustainable. And in, I would say, the current federal climate, it makes it very anxiety provoking. And I think the other big challenge is always time management, and especially for clinician scientists, but really for anybody, we have to protect our time. So I've tried to protect Thursdays so I never operate. I've still operated less than 15 times in the last 16 years on a Thursday, which I'm proud of. So most of Thursdays are protected. Some Fridays are protected. And I think it helps that the science is my favorite thing to do. So I try to devote extra time. It's going to be a little bit clinical reading, but a lot of science reading and engaging with lab, talking with lab colleagues, people, following up on things that I should. But I think the main things are always going to be the same for every scientist. How do you find more time in the day? How do you find more funding to do the things that you want to do? I think it's all about the process because, in general, most of our scientific questions we're going to end up with failures. So when we have a hypothesis, it's literally a guess. And I think a lot of times experiments don't work, grants don't get funded. I remember being told that a successful grant writer gets 15% of grants scored. And I was like, wow, I've never been in the 15th percentile. That sounds really scary, but that's our baseline is we're going to get most of the things that we do rejected, and then you just have to be able to bounce back. But I think it's really understanding that it's all about the process and you can't subvert the process. You're trying to figure out how things work, why they work the way they do. Any shortcuts aren't going to help. You can learn how to write a better grant, but really it's about explaining your grant better. It's about getting your paper written in a way that reviewers are going to understand, and you can't shortcut the science. Um, so basically, be resilient, understand that it's not good science is about resilience and figuring out the right answer, not figuring out the answer that you think you want it to be, because that's how you end up taking shortcuts and going down the wrong path and ending up being frustrated. I'm hopeful that it allows our lab a little bit more scientific clout in study section, because I think one of the challenges we have is when we get our grants reviewed, we're being read by immunologists and dermatologists and not always orthopedic surgeons that see the value of our work. Um, but I think it's mainly just an honor that I was fortunate to be able to share with my colleagues, and in particular Shui Liu, who I've worked with now for 15 years. And winning it as a surgeon scientist, hopefully allows other orthopedic residents and junior faculty to decide that they want to apply for things like this, be aggressive about pursuing research. It was great seeing that this was essentially a almost all sports medicine group that won, seeing Matt Provencher and Brian Cole at the same time. And then seeing Nico Puzzi from the Cleveland Clinic win was great too, because his research is entirely different than a lot of the basic science stuff, but it's really transformative in how it's changed our thinking about hip and knee replacements, which is frankly probably the most important surgery we do in orthopedic surgery. The ORS has been very supportive. I think it's a great meeting. I love the balance between spotlight talks and just straight up science, and then they have really good poster sessions. So the combination of having like respected leaders come give an overview, plus having that ability to see relatively short talks and understand what cutting edge is, and then kind of then go and talk to people at the posters, I think has been really informative. I also think the fact that it's really inclusive. A lot of meetings like AOSSM, for instance, it's really hard to get a poster or a presentation in, and there's a lot of instructional courses, but you don't get that opportunity to present. So being able to get the entire lab, if they want something to present, they can present. I think that's really important. And I think being able to work with some of the members of the ORS, getting to know a broader scientific community, getting to work with Matt Zuleg, who I think pretty much runs ORS, is great. In terms of my favorite thing about it, yeah, I think the fact that the meeting runs well, efficiently, tends to pick pretty decent cities, and is always, every few years, the meeting gets a little bit better than it was a few years before. I think when I think back to would I have done designed experiments differently, written a grant a little bit better, given a talk better, especially talks early on, I wish I'd given a lot smoother. I can definitely think of things that I would have done differently in a small scale, but large scale, I don't think I would have changed anything. I love the combination of having clinical stuff to do on some days, basic science to do on the others. I love my colleagues at UCSF. I love the collaborations we have. So I don't think I really would have done anything differently other than the same things that we all should be fine tuning. We did a session about 7 or 8 years ago in a leadership forum where they ask you to like give a presentation, and then you're supposed to be clapped at for 30 seconds and just sit up there and take it. And it's very awkward. And I think scientists in general, we tend to remember our failures and then forget the things that we did well. And so I think I don't really remember the moments that I got grants or the moments that I opened emails and was like, oh, I won something. But I think a lot of little things, mainly from other people in the lab. I still remember the first time we had an undergrad present an oral presentation, and I remember who asked him a question, what that question was, and how that was changed once he said, I'm just an undergrad. I don't really know. The first time we had a high school present at ORS. And then the main thing is seeing the people that we've really brought through the system. So seeing like Mike Davies and Steve Garcia, who were med students with us, then they were residents with us. They did time in the lab. And now Mike is a colleague and he's on faculty. Stephen Garcia is going to be on faculty. So seeing those people grow up. It's also, been the other thing I'd have to point out has been really fun to publish with both of my kid or two of my oldest kids. Um, and then that just means the other three are not yet ready to, but I've been able to write things not necessarily on the basic science front with the oldest two. And that's been really nice and kind of full circle because I still remember my dad letting me be an acknowledgement when I helped him make tables with an early version of Excel when I was in high school. So I feel like that was at least a full circle moment. And once I get all five kids on one paper together, then I feel like that'll complete it. I like to surf when the surf is decent, with climate change is a little bit less and less, but we have excellent surfing in Northern California. I love to hang out with my family. One of the great things about having five kids is there's always someone to hang out with. I like to hang out with my wife, Gail, because we have a great relationship. We exercise together, we read together. And then I have a sports medicine podcast that is actually a lot of fun to do with some of my colleagues, and we get some very interesting and eclectic guests on. So that's an interesting way to get different people interested in sports medicine and then hear their perspectives on sports medicine, things like pain and postoperative recovery, what it's like to be a stunt double for Chris Hemsworth. We get all sorts of random people on it. So that's been a lot of fun.